Cells of immune system. There are two main types of cells involved in the working of immune system. They are lymphocyte and antigen presenting cell. Lymphocytes are the main cells of the immune system. They, like other blood corpuscles, arise from the stem cell, the hemocytoblast present in the liver of the fetus and in the bone marrow in adult. Some of them undergo differentiation in the gut, associated bursal lymphoid tissue, tonsils, Peyer's patches, and are called bursal or B lymphocyte. Others are differentiated in the thymus gland and are termed as T lymphocyte. The mature lymphocyte pass into the body fluid, blood and lymph and circulate in the body. Many of them stay in the lymph nodes. The B lymphocyte and T lymphocyte forms humoral or antibody-mediated immune system, AMIS, and cell-mediated immune system, CMIS, respectively. Both the immune system needs antigen to come into action, but they respond in different ways. Mechanism of response of T lymphocyte to antigen. On coming in contact with an antigen, T lymphocyte forms clones of T cells, which are similar but they perform different functions. The clone has four types of lymphocyte: helper T cells, killer T cells or cytotoxic T cells, suppressor T cells, memory T cells. Helper T cells. Sensitized helper T cells produce lymphokines for performing several types of functions like proliferation of other T cells, stimulation of B lymphocyte, macrophages. Killer T cells or cytotoxic T cells. They directly attack and destroy invading microbes, infected body cells and cancer cells. Killer T cells binds to infected cells and secrete perforins. Then perforins forms a hole in infected cell. It also releases substances that kills the cell, hence the name cytotoxic T cell. Suppressor T cells. These cells suppress entire immune system against attack on the own body cell. Memory T cells. These are previously sensitized cells which retain the sensitization memory for long time in the future. Mechanism of action of B lymphocyte to antigen. B lymphocyte are sensitized directly by both antigen as well as by helper T cells. Activated B lymphocyte multiplies very fast to produce clonoplasma cells and memory B cells. The plasma cell produces specialized glycoprotein called antibodies, which are circulated through body fluid, humor, like blood and lymph. The antibody molecules may bind to a cell membrane or they remain free. The free antibodies have three main functions: agglutination, opsonization, and neutralization. Agglutination of particulate matter including bacteria and viruses. The immobilized mass is then engulfed by phagocyte. Opsonization or coating of bacteria to facilitate their subsequent phagocytosis by macrophages. Neutralization of toxin released by bacteria. Example, tetanus toxin. Each antibody is specific to a particular antigen. Antigen presenting cells. They engulf invading pathogens and process the antigens. The processed antigens are presented on their own surface. These cells are able to deliver a stimulatory signal that is necessary for activation of helper T cell.